Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. So in the last lecture I was concentrating on lactic acid production. So I talked already about the basic aspects of lactic acid. Now I shall show you how industry that produces lactic acid. So if you look at the raw materials that is used most suitable for lactic acid production is the whey, because whey is the byproduct of the dairy industry. Because you know that when we produce this cottage cheese, that you know that whey comes out, that this contains very less amount of protein and fat, but the liquid mostly contains the lactose, and which is a very good raw materials for the lactic acid formation. The whey, why we use the whey for this fermentation process? The reason is that it is byproduct of the dairy industry, and second is that it is a very cheap raw materials. It contains 4.6% to about 7% of lactose, then 10 to 15% of sugar, because the cream is removed from the milk and casein is precipitated from the use of hydrochloric acid, or and also sometimes we use in the house we use the calcium lactate to precipitate out the protein. When the whey which is the product left after the separation of the cream and the casein from the milk contains albumin, approximately 4.56% of lactose, vitamin G, mineral salts, and the water. Because you know that this solid material mostly it contains the milk, protein and fat, and all the minerals and the vitamin G and lactose goes in the soluble forms. This media serve as nutrient substrate for the manufacture of lactic acid. Now let me show you the steps involved in the lactic acid production. It's very because we prepared the culture in a slant, then from the slant we inoculate in the media in the sec flux, where the liquid media, that this is the solid media, then this is the agar slant we grow the culture, then in liquid media in the sec flux we grow this culture. After that it grow in a control fermenters, and we get the cell mass, and this cell mass we put it in the production fermenter, which produces lactic acid, but it pass through the different downstream processing like filtration, evaporation and the purification. So this is the so three steps involved, major steps. One is preparation of starter. Starter means the organism that we prepare for this production, and there is fermentation, that what is carried out in the production fermenter, and the lactic acid. So if you look at the normal that fermentation industry, whatever we have, it is almost similar to that. In the normal fermentation industry also, I told you that every industry they have two reactors, bioreactors, one for the cell mass production, another for the product for the cell mass production we call it inoculum vessel, and production fermenter we call it PF, the production fermenter. So now let us see that what is the first step that we have in the preparation of a starter. A quarts of sterilized skim milk is inoculated with the culture Lactobacillus delbrueckii containing yeast that causes the fermentation to become more vigorous. After incubation for 24 hours, because you know that one thing here I want to point out, that doubling time of bacteria is much less as compared to yeast cells. If you add, it gives some kind of nutrient to the fermentation broth. It does not affect the growth of the organism to a great extent. I told you can remember in the citric acid fermentation process, we use the cane molasses as the raw material for the production of citric acid. And main culprit, contaminants that we have in the citric acid fermentation process is the yeast. Why? Because the cane molasses is the best raw material for the growth of yeast cells. And if you look at the doubling time of yeast cell is much less as compared to that of fungi. So, if little yeast enter into the system, that will not allow the fungi to grow. So, here we do not have any problem if you use this because your bacteria, you look at the yeast, if your bacteria doubling time is much less, lactobacillus delbrueckii and contains yeast causes the to become the vigorous. Then after incubation for 24 hours at temperature 43 degrees centigrade, the content of the bottle is placed in a container with pasteurized skim milk. This content of the jug transferred after the inoculation 43 degrees at 24 hours into a glass lined steel tank containing 500 gallons of pasteurized whey. Because that one thing I want to point out that lactic acid has a corrosive property. So, the special type of construction material of construction of the fermentation the tank is to be maintained. Following incubation at 43 degrees centigrade for 24 hours, the tartar is added to the main fermentation tank. So, we first prepared the culture, what we call inoculum is the inoculum, and then this inoculum we use to the production fermenter. The step 2 is the material of construction. The lactic acid is highly corrosive as I pointed out, and therefore, it has problem in relation to its production, processing and handling. The non-corrosive material is usually used for the construction of fermenters. The two of the material of construction which is mostly resistant to lactic acid are silver and tantalum. This is the two metals with their quite resistant to lactic acid. So, this is usually recommended. The group of materials which are fairly resistant to lactic acid are nickel, inconel. Inconel is nothing but nickel chromium based superalloy and low iron alloy. Then group materials which are poor resistant to lactic acid, they are iron, copper and chrome steel, etc. I mentioned that chromium plays important to increase the corrosive property of the stainless steel, but here it is not that effective. And usually the fermenter is constructed with heavy wood, because in the acetic acid fermentation process I was talking about the beechwood saving, which is a soft wood with a lot of pore inside. But here we are talking about the hardwood. Hardwood also like oak wood we sometimes we can use for this as a vessel, but hardwood we can use as a vessel. Modern fermenter is constructed with stainless steel. Fermenter is provided with the agitator. Agitator is required so that cell can remain in suspension. The inoculated whey is maintained as 43 degrees centigrade. Fermentation time is 42 hours. Calcium hydroxide in the form of slurry is added to in the fermentation mass every 6 hours to keep the acidity much below 1%, 0.1%. Because if you use the high, you allow the acidity build up, then that will hinder the growth of the organism. So, this is to be maintained. So, we add and I already mentioned in case of citric acid fermentation process how calcium hydroxide is produced. I told you that calcium carbonate, which is present in the rock, this when you heat it produces calcium oxide and carbon dioxide. The carbon dioxide will goes out. Then this calcium oxide in presence of water, you make a slurry, you will get calcium hydroxide. This calcium hydroxide you have to add. that you have to add. And when you add the calcium hydroxide, when in presence of lactic acid, it produces calcium lactate. So, neutralizing the acidic lactic acid with lime has several advantages, such as the shorter fermentation time, higher yield, prevention of bacterial inhibition due to the production of acid. So, these are the reason why we use the lime. Now, this is the flow diagram of the process. This flow diagram is like this, that we take the cheese whey, we can take whey from other source also. Then we do the ultrafiltration, whey proteins we take it out, maybe you heat it, then protein will be precipitated out, then you can take out the whey protein through the ultrafiltration. Then in the fermenters we inoculate the lactobacillus lactic acid bacteria here. Then after production we pass it to the nanofilter, we get 2 to 10% of lactic acid. Then RO filtration, the reverse osmosis that we carried out to get the higher concentration of lactic acid. Then vacuum evaporation, we get 90 to 98% lactic acid, and this lactic acid we can go for the polymerization reaction to get the polylactic acid. So, what you call polylactide, because I told you that polylactic acid has tremendous potentiality in the pharmaceutical industries. Now, I want to show you the reactions how lactide formation is there. This is the lactic acid. If you look at 2 lactic acid, when formed, it is from the lactide, and this lactide actually help for the formation of polylactic acid. Now, if you look at the polylactic acid is biodegradable. Why we use the polylactic acid? The reason is that it is biodegradable, and it is mostly used in food handling and medical implant that biodegradable within the body over time. That this is very important, medical implants that biodegrade within the body over time. So, this is why it is used for the medicinal purpose, or particularly in case of surgical purpose is largely used. Like most of the plastics, it has potential to be toxic if inhaled or absorb into the skin, eyes as a vapor or liquid. During manufacturing process, obviously, if this polymer equally affected to the human health, it is no good, but when it is solidified, it does not have bad effect for the human health. It is easily biodegradable. So, then the question comes how we recovered the lactic acid. Calcium hydroxide is added to the broth to lower the acidity below 0.1%, and then heated to 80 to 95 degrees centigrade with the help of coagulation of protein and filtered. The filtered then allowed to evaporate to dryness to get the calcium lactate. And so, initially our intention is to produce the calcium lactate, and this pure calcium lactate, it is dissolved in water and treated with the decoloring carbon and mix it through thoroughly and stand for 15 minutes and filter, because it may give some kind of color to the product. That is why we use the activated carbon. Just to activated carbon, I mentioned before also, it has some kind of bleaching property. So, if you use the color can be removed, you have and then we prepared the lactic acid calcium lactate to treat it with H2SO4 to get the solution of lactic acid, and calcium sulphate get precipitated out. We can separate out the, like in case of citric acid also, we hydrolyze the calcium citrate by H2SO4, we separate calcium sulphate in the form of gypsum filter. In the gypsum filter, we separate this calcium sulphate to H2O, that is called gypsum, which is a very good raw material for the cement industry. and then the dilute solution is heated to high temperature, then some chemical changes may occur and it forms the calcium anhydride. So, if you use the very high temperature, it again you will get the different product, the lacto, which is not desirable. Now, let us see that how actually lactic acid is produced. This is the fermentation product. After that, you will get this, and you get the calcium lactate about 10% weight by volume, and when it goes to the reactor where you push the this H2SO4 and heat it for 30 degrees centigrade. Now, and then we in previous case I mentioned that we heat it to 80 to 90 degrees centigrade, and then we centrifuge it at high temperature. We get calcium sulfate, and then we evaporate it so that your concentration of lactic acid increases. We filter it, impurities will goes out, lactic acid equals 60% weight by this. This is available in the liquid form. This lactic acid, the reaction is like this: calcium lactate plus H2SO4 give lactic acid and calcium sulfate. Now, containers used in distribution. Now, question comes, since lactic acid has highly corrosive property, so distribution in the container also is very important. The aqueous solution of lactic acid is sold and distributed in wooden or paraffin lined barrels of oak, cypress and pitch pine, or in line car tanks. The higher grade acid are solid in glass line in cars or carboys. So, this is some glass line materials also can be used for that, for transferring this material. Then it will not affect the material of construction to a great extent. Otherwise, you have to do otherwise the material will be affected with the help of lactic acid. So, this is very important. Now, let me tell you that what are the different types of lactic acid usually available in the market, because this is also very important. The lactic acid can be available in the different forms. One is crude or technical grade lactic acid, which is 20 to 80%. This is largely used, this lactic acid strength is there. Edible, that is 50 to 80% that we have lactic acid. Plastic, we required 50 to 80% lactic acid. In the United States, pharmacopoeia, that is UPS medicinal grade, is 85%. So, different percentage of lactic acid that is used for different purpose that we have. Now, if you look at what are the different grades that we have that uses for different purposes. As for example, crude grade, this is colored, the varies from differ from yellow to brown, and it is used in deliming hides. Limes are removed that animal skin. This is particularly I can tell in the tannery industry is largely used for that. And then dyeing the silk and other textiles, that is the crude grade of lactic acid is largely used. Then edible, usually this is the yellow color, and that is used in the jam, jelly and brine fermentation, casein precipitation and for fat extraction. So, that different purpose it is used for edible. When we use, I can give the example of calcium lactate. I was telling, the largely we use in the house for precipitating the milk, protein and fat, because we use for several purpose. Then we have other different grades. We have plastic grades. It is colorless and generally used for the manufacture of plastics, because I told you that lactic acid has huge market potentiality in the pharmaceutical industry, and this plastic grade has lot of use in this industry. and UPS is a pharmaceutical grade of the lactic acid and used in the pharmaceutical formulation purpose. So, this is all about the lactic acid fermentation process. Now, here I want to summarize like this, that lactic acid is produced by using two different type of bacteria in the industry largely used. One is Lactobacillus delbrueckii, another Lactobacillus bulgaricus. The temperature is little bit high, usually 43 degree centigrade or higher temperature that is used for this fermentation process. And raw materials contains, we find that whey, which is the byproduct of the dairy industry. It is very fine, suitable for this fermentation process, and it has been observed that amino acid present in the yeast extract and malt extract, they find very suitable for the growth of the yeast cells, growth of the lactic acid bacteria. Then when you come to the fermentation process, that during the fermentation process, we maintain the acidity by using calcium hydroxide or calcium carbonate. And since we are adding the calcium hydroxide will react with lactic acid and form calcium lactate, and then after this required about 42 to 46 hours, and after fermented your inoculum fermentation for the cell mass production is required 24 hours, and then after the production fermented, the material we treated with we concentrate that, we separate out the calcium lactate, and then we hydrolyze in presence of H2SO4, we get the lactic acid and calcium sulfate precipitated out with the help of filtration. Other cases also we do some kind of filtration process just to separate out lot of suspended material in the fermentation broth, including the bacterial suspension. So, this is all about the lactic acid bacteria. Thank you very much.